Welcome back to the deep dive. Today we are really getting into the weeds, cracking the code on the body's chemical messaging system, the endocrine system. We're not just listing hormones. We want to understand the molecular nuts and bolts. How fast do they work? How long do they last? What makes them tick? We've got some great source material here, almost like a blueprint. It lays out the different hormone types, how they're stored, released, and crucially, the signaling pathways inside the cell that really control everything.
That really is the core mission today, isn't it? Moving beyond just names. Like, why is epinephrine so incredibly fast while thyroid hormone takes, well, ages, relatively speaking? The aha moment, I think, comes when you realize the cell uses completely different tools, different receptors, different messengers, depending on the job at hand. Is it an urgent message or a long-term plan?
Right, and maybe the biggest place to start is storage. Is the message preloaded and ready to fire, or does it have to be built from scratch every single time?
That storage question is fundamental, and it all ties back to solubility. Can it cross that fatty cell membrane or not? That really divides our three main classes.
Okay, let's start with the first group then. The peptide and protein hormones. These are the big players, right? Insulin, glucagon, growth hormone, the fast actors.
Exactly. They're water-soluble. Good news for traveling in the blood, but bad news for getting into cells.
So they hit a roadblock at the cell membrane.
Precisely. But their production is geared for speed. Made on ribosomes, processed, and here's the key, stored in huge amounts in vesicles, like little grenades ready to go off.
Grenades, I like that.
Yeah. And since they can't get in, they have to knock on the door, use cell surface receptors, GPCRs, or RTKs. They rely on internal helpers, second messengers, to carry the signal inside. That's why the response is so fast, seconds to minutes. It's like sending a loudspeaker announcement.
Okay, so that's the fast crowd. Now let's completely flip the script. Steroid hormones, cortisol, the sex hormones, derived from cholesterol, so they're super lipid soluble.
Yeah.
How does that change the game?
Oh, it changes absolutely everything because they can slip through the membrane. They don't need storage. They're made completely on demand.
No vesicles, no pre-packaging.
None. Synthesize it and it just diffuses right out. And its receptor, it's waiting inside the cell, cytoplasm or nucleus. Their whole purpose isn't speed. It's about deep, lasting changes. They directly mess with gene transcription.
Turning genes on or off.
Exactly, which, inherently takes time. That's why their onset is slower, half an hour, maybe hours. But the effects stick around.
And then there's the third category, kind of the odd one out, thyroid hormones, T3 and T4. They come from amino acids, but they act slow and are lipid soluble, like steroids. But our sources point out a really critical difference in their mechanism.
Yeah, the thyroid system is quite elegant, actually. It's lipid soluble, finds its receptor inside the nucleus. But unlike steroids, that receptor is already sitting there, bound to the DNA.
It's already parked on the specific DNA site, the thyroid response element, or TRE.
Right on the TRE, usually paired up with another receptor called RXR. It's just waiting. Often, it's actively silencing the gene with a core-pressor protein attached.
So T3 arrives.
And it doesn't need to find the parking spot. It just causes this swap. The core-pressor gets kicked off, and a coactivator protein jumps on. The whole machine is pre-assembled.
So it's less about building the factory and more about just flipping the on switch for the machinery that's already there.
That's a great way to put it. And that pre-assembly contributes to its very slow onset, but also its incredibly long duration. We're talking days.
Okay, so these hormones get released, but their journey in the bloodstream isn't straightforward either, especially for the lipid-soluble ones. What's going on there?
Right, solubility again. For steroids and thyroid hormone, almost all of it, like 99% plus, gets grabbed by carrier proteins in the blood. Think of things like thyroid-binding globulin, TBG, cortisol-binding globulin, CBG, even albumin.
So only a tiny fraction is actually floating free.
A tiny, tiny fraction, less than 1%. And that free hormone is the only part that's biologically active, the only part that can actually slip into the cell and do its job.
And the bound hormone acts like a reservoir, protecting it.
Exactly. It's a buffer, a reservoir, and it protects the hormone from being broken down quickly by the liver or filtered out by the kidneys. That's why the half-lives are so wildly different.
How different are we talking?
Massive range. Epinephrine, a free peptide, gone in seconds. Other peptides, maybe minutes. Thyroid hormone, because it's so heavily bound, its half-life is measured in days.
Wow. Okay, and that bound versus free concept is super important clinically, isn't it? Let's take the pregnancy example from the sources. Pregnancy boosts TBG levels. What happens then?
So you measure the total T4, all the T4 bound and free, and it'll be high because there's more TBG to hold on to it.
But the patient isn't actually hyperthyroid.
Nope. The body adjusts. The active free T4 level stays right in the normal range. The thermostat, so to speak, is working fine. Clinically, they're euthyroid. You have to look at the free T4.
Got it. And the flip side, someone with, say, liver failure or nephrotic syndrome where they're losing protein, including TBG.
Then you see the opposite. Less TBG means lower total T4, but again, the body compensates, and the free T4 concentration usually remains normal. The bottom line for these lipid-soluble hormones is always check the free fraction. That tells you the real story.
Okay, really clear. Let's switch back to the speedsters, the peptides and catecholamines, using those G protein-coupled receptors, the GPCRs. Why this reliance on internal second messengers?
Well, because the hormone itself, the first messenger, can't get in, so the receptor on the surface needs an inside helper. It activates a G protein, which then turns on an enzyme or opens a channel to produce these small molecules inside the cell, the second messenger.
And the big advantage is amplification.
Huge amplification. One hormone molecule binding outside can trigger the production of thousands, even millions, of second messenger molecules inside. That's how you get such a rapid, strong response from a tiny amount of hormone.
Our charts lay out the main G protein flavors, GS, G, and GQ. Let's start with G stimulatory. Think TSH, glucagon. What's the chain reaction?
GOS gets turned on, it bumps into adenyl cyclase, turns that enzyme on. Adenyl cyclase then starts churning out cyclic AMP, or CAMP.
KMP, the classic second messenger.
Classic. And increased CAMP levels activate protein kinase A, or PKA. PKA is the workhorse enzyme that phosphorylates other proteins to change their activity, causing the cell's immediate response.
Wait, PKA can also affect genes. That seems less immediate. How does that work?
It bridges the gap. Active PKA can actually travel into the nucleus. There, it phosphorylates a transcription factor called CREB.
CREB, right.
Phosphorylated CREB binds to specific DNA sequences called CRE's CAMP response elements, and that influences which genes get turned on or off over the longer term, so you get both. Fast cytoplasmic effects and slower nuclear effects from the same pathway. CMP to PKA to CREB.
Clever. Okay, then there's G inhibitory. Does the opposite. Shuts down adenyl cyclase, lowers CMP. Simple enough. What about the third one, GQ, used by angiotensin II, for instance?
GQ takes a different route. It activates an enzyme called phospholipase C, or PLC.
Okay, PLC, what does that do?
PLC chops up a specific membrane lipid, PIP2, into two second messengers, IP3, a inositol triphosphate, and DAG, diacylglycerol.
2 for the price of 1.
Kinda, yeah. IP3 floats over to the endoplasmic reticulum and triggers a big release of stored calcium into the cytoplasm. Calcium itself is a major second messenger.
A day.
DAG stays in the membrane and, along with the released calcium, activates another kinase called protein kinase C, or PKC. So GQ gives you calcium signals and PKC activation.
Right, these pathways fire up incredibly fast.
Uh-huh.
How does the cell slam on the brakes just as quickly?
Couple of ways. The G protein itself has a built-in timer. The G-alpha subunit slowly hydrolyzes its bound GTP back to GDP, turning itself off. There are even helper proteins, GAPs, that speed this up.
Okay, so the G protein switch turns off. But what about the second messenger, the KMP itself?
That's crucial. There's an enzyme called phosphodiesterase, or PDE. Its whole job is to break down KMP into an inactive form, 5-AMP. It's constantly cleaning up the signal.
And inhibiting that enzyme is a big deal pharmacologically, right? Like caffeine.
Yes, exactly. Caffeine and theophylline are PDE inhibitors. They block the breakdown of KMP so the signal lasts longer. That's part of how they work as, say, bronchodilators. Same idea with drugs like sildenafil. It inhibits a specific PDE for cGMP, leading to vasodilation. Blocking the off switch keeps the signal on.
Fascinating.
Huh?
Okay, let's shift gears to the other major fast receptor type. Receptor tyrosine kinases, or RTKs. Insulin is the classic example, and our sources say its receptor is kind of unique.
It really is. Most RTKs only come together as a dimer when the hormone binds. The insulin receptor, though, it's already hanging out as a dimer, 2 alpha subunits outside, 2 beta subunits crossing the membrane even before insulin shows up.
So insulin binds to this preformed dimer. Then what?
It triggers autophosphorylation. The intracellular parts of the beta subunits phosphorylate each other on specific tyrosine residues. The receptor itself is the kinase.
A tyrosine-specific kinase activity built right in.
Precisely. And those newly phosphorylated tyrosines act like docking sites. They recruit other proteins, initiating signals downstream. And for insulin, there are two main pathways you absolutely need to know.
Okay, path number one.
The PI3 chiactypt pathway. This is the metabolic arm of insulin signaling. It's responsible for getting GLUT4 transporters to the cell surface to take up glucose for stimulating glycogen synthesis, and critically, for suppressing fat breakdown, lipolysis, and ketone body production, ketogenesis. Lack of this suppression is why you get diabetic ketoacidosis, DKA, and type 1 diabetes, unchecked lipolysis.
Got it. Metabolic effects via PI3 chiactypt. What's the second major pathway?
That's the MAPK pathway. Think mitogen-activated protein kinase. This one is more about the growth and proliferation effects of insulin. It's a more complex cascade.
The key step.
It involves activating proteins like RAS and RAF, leading down to a kinase called MEK. MEK is interesting because it phosphorylates the next kinase, ERK, on both a threonine and a tyrosine residue. Activated ERK then goes on to influence gene transcription related to cell growth.
So two distinct signals from the same receptor, metabolic and mitogenic. How does the soul turn the signal off?
Well, you have phosphatases that remove those key phosphate groups, reversing the activation steps. But a really important mechanism, especially for long-term regulation, is receptor endocytosis.
The cell just pulls the whole receptor inside.
Yeah, it internalizes the receptor hormone complex. Then it can either degrade it in lysosomes or recycle the receptor back to the surface. This process of pulling receptors off the surface is a major way the cell dials down its sensitivity to insulin resistance.
Right, that makes sense. Okay, before we wrap up, I want to loop back to that GQ pathway, PLC making IP3 and DA. There's a huge connection here to inflammation and pharmacology, a really high-yield area in our sources.
Absolutely. So GQ activates PLC, PLC makes DAG. We need to follow that DAG molecule. It can be acted on by another enzyme, DGAG lipase, to produce arachidonic acid, or AA.
The arachidonic acid, okay, that name rings bells. That's the starting material for a lot of inflammatory stuff.
Pretty much all the major players. AA is the critical precursor. It's the substrate for two major enzyme pathways that generate inflammatory mediators.
Which are?
First, cyclooxygenase, COX1 and KOX2. These enzymes convert AA into prostaglandins and thromboxanes. Think pain, fever, inflammation, blood clotting.
Okay, COX pathway. And the second?
5-lipoxygenase, or 5-LOX. This enzyme converts AA into leukotrienes. These are major players in allergy and asthma, causing things like bronchoconstriction.
So AA is this central hub feeding into both prostaglandins, thromboxanes via COX, and leukotrienes via 5-LOX. That makes it a prime target for drugs.
Exactly. And this is where you see the crucial difference between how NSAIDs and steroids work.
Okay, lay out for us. NSAIDs, first aspirin, ibuprofen.
NSAIDs work by blocking the HEOX enzymes, EOX1 and or KOX2. They stop the conversion of AA into prostaglandins and thromboxanes. But importantly, AA is still being produced, and the leukotriene pathway is untouched.
Right, they block one specific outflow path. Now, steroids like prednisone, where do they hit the system?
Steroids work much further upstream. They don't block COX or LOX directly. Instead, they cause the cell to produce more of a protein called lipocortin, also known as annexin 1.
Lipocortin. What does that do?
Lipocortin inhibits phospholipase A, or PLA, and PLA is the main enzyme responsible for releasing arachidonic acid from membrane lipids in the first place.
Whoa. So steroids stop the production of the raw material, AA itself.
Precisely. They shut down the supply line for both the COX and the 5-LOX pathways. By preventing AA release, they block the synthesis of prostaglandins, thromboxanes, and leukotrienes.
That's why they're such broad-spectrum, potent anti-inflammatories. They're not just plugging one hole, they're turning off the tap at the source.
That's the key distinction. NSAIDs block downstream enzymes. Steroids block the upstream liberation of the precursor AA by inhibiting PLA via lipocortin. It's a fundamental difference in mechanism.
Fantastic. Okay, that was quite the journey through the molecular signaling world. Yeah. We really saw the stark contrast. The peptides stored, fast, water-soluble, using surface receptors and second messengers, versus the steroids and thyroid hormones made on demand. Slow, lipid-soluble, going right for the nucleus.
And it all comes down to the architecture, doesn't it? The incredible speed and amplification you get from GPCRs and RTKs compared to the deep gene-level changes from nuclear receptors. It allows for such diverse control. Think about epinephrine using GQ via alpha-1 receptors for one effect, like vasoconstriction, and Gs via beta receptors for a totally different effect, like bronchodilation. Same molecule, different cellular machinery.
Amazing versatility. So, a final thought for everyone listening. We touched on receptor internalization for insulin, pulling receptors off the surface to turn off the signal quickly. But think about that same process playing out over weeks, months, years. The cell constantly adjusting how many receptors are on its surface, degrading them faster or recycling them slower. How does that chronic up or down regulation change the cell's entire sensitivity to a hormone? That molecular detail, that constant tuning of receptor numbers, is exactly how long-term changes like insulin resistance or tolerance to a drug can develop. It's how these tiny signaling events build up to impact your overall health. Something to chew on.